We welcome you all to the International Conference cum Workshop Global Summit on Innovative Drug Design, Discovery and Translational Research.
I would now request all the dignitaries to mark the beginning of this 3 days conference cum workshop by lighting the lamp.
Respected Honorable Vice Chancellor Professor Prakash Babu, respected Professor Dinakar Samanki, Director ICGEB, New Delhi, Professor Basan Kumar Tiwari, Dean in Charge, Professor Dinakar Rao, Convener of the conference, Professor Sanjeev Kumar Singh, General Secretary, BITS, Organizing Secretary Dr. Mohan Kumari, distinguished invited speakers, professors, participants, scholars and students, ladies and gentlemen. Very good morning to everyone. It is a great pleasure and honor to give a welcome address on the occasion of the International Conference cum Workshop or Global Summit on Innovative Drug Design, Development and Translational Research conducted by the Department of Bioinformatics, Pondicherry University. This department is one of the best departments in the country and recognized as a center of excellence in bioinformatics by the Department of Information Technology, Government of India. And this department is running 4 independent programs, PhD in bioinformatics and MTech computational biology, which is the students are selected through Gallery. They are getting a 12,000 fellowship per month for 2 years. And MSc Bioinformatics, that's a UGC innovative program, and BSc Honors Bioinformatics, that is a NEP program. We are also involving a BSc Honors Life Sciences teaching program also. So this is a very big department, approximately 240 students with 12 faculties. The strength of the department is the high quality research publications with 100% placement of students either in industry or in academic institutions. I'm really very happy. First of all, I'm really very happy to welcome our Honorable Vice Chancellor in this conference. Sir, thank you very much. You are always giving a constant support and encouragement to this department, sir. And on this occasion, I take this opportunity to congratulate our Honorable Vice Chancellor to upgrade this Pondicherry University to NAC A to NAC A+, sir. You have done well done, sir. And because the university was struggling for last 15 years with the A grade, and you made it done within two months, sir. Thank you very much and well done, sir. And we are very happy to welcoming you in this conference and thank you for accepting and giving the presidential address in this conference. And also, the Department of Bioinformatics, along with the BITS Society, very honored and happy to hear your BITS Lifetime Achievement Award lecture, sir. Thank you very much. I am also happy to welcome Professor Dinakar Samanki, former director, ICGEB, so to accept and give the inaugural address, sir. Thank you very much. I'm also happy to welcome our Dean in Charge, Professor Basan Kumar Tiwari, accepting and giving a felicitation address. I'm very happy to welcome our Dinakar Rao, convener of the conference, to give the scope of this seminar. I'm happy to welcome the General Secretary, BITS Society, Professor Sanjeev Kumar Sharma, accepting and giving the special address. I'm also happy to welcome our Organizing Secretary, Dr. Mohan Kumari. I'm also happy to invite all distinguished invited speakers, professors, participants, students and scholars, and all into the conference. Thank you very much, Nandisi.
Thank you so much, sir. I now request Dr. Dinakar K. Salunke to felicitate Professor P. Prakash Babu.
Honorable dignitaries on dais, of the dais, faculty, participants, and dear students. I extend warm welcome to you all to the International Conference cum Workshop, Global Summit on Innovative Drug Design, Discovery, and Translational Research. As you know, drug discovery and development stands as pillars of innovation and progress. From combating infectious disease to address chronic conditions, the quest for new therapeutic agents drives scientists, clinicians, and pharma companies worldwide. Early civilization relied on natural substances such as plants, animals, and minerals, plants extracts for medicinal purpose, often based on empirical observations and folklore. Today, drug discovery encompasses a multidisciplinary approach that integrates knowledge from chemistry, biology, pharmacology, and computational sciences. The process typically begins with target identification, wherein researchers identify a specific molecule, biological pathways implicated in disease pathology. This may involve genomics, proteomics, and other omics technology to elucidate disease mechanisms and identify druggable targets. Once a target is identified, the next step is lead identification, whereas researchers screen compound libraries, design molecules with potential to modulate targets activity. Nowadays, the drug discovery and development is constantly evolving. Emerging technologies such as high throughput screening, chemical space docking, and artificial intelligence are revolutionizing the drug discovery process, enabling faster and more efficient identification of novel therapeutics targets and candidates, and successfully moving towards bench to bed and lab to land. So, I would like to express my sincere gratitude to the distinguished invited speakers for their presence and all sponsors for their contribution to this conference. Finally, I sincerely hope that participants will benefit from the technical and scientific content of this conference, particularly young researchers. I wish you all very successful conference and enjoyable stay in Pondicherry and Pondicherry University. Pondicherry University is a NAC A+, lush green and eco-friendly university. I request all participants visit departments and interact with faculty for further collaborations. So, I wish a conference a grand success. Thank you very much.
Thank you, sir, for presenting such an apt description behind the idea of the conference. Now I request Professor Basan K. Tiwari to address the audience.
Good morning, respected Honorable Vice Chancellor, Professor Prakash Babu, respected Chief Guest of today's program, Dr. Dinakar M. Salunke, my colleagues on the dais. I welcome all of you on my behalf as a part of Department of Bioinformatics and also in the School of Life Sciences. This school consists of 7 departments and it's a very dynamic school, and we also have a campus in Port Blair where we take care of the marine biology department. So we have a very good interaction among the departments as far as teaching is concerned and research as well. In teaching, our Department of Bioinformatics provide some basic courses for our sister department in School of Life Sciences as well as School of Physical Sciences like chemistry. Students also take up our courses. So it's a great privilege for me to welcome you all on this occasion, and I also thank my colleagues Dr. Dinakar Rao and Dr. Mohan Kumar for taking their efforts to make this program success. Thank you very much.
Thank you so much, sir. I now call upon Dr. Dinakar K. Salunke to give the inaugural address.
Good morning to you all. Vice Chancellor of the Pondicherry University, distinguished colleagues on the dais, students and friends. It is actually great pleasure to be in Pondicherry to attend this global summit on innovative drug design, discovery and translational research. I missed coming about 2 months back when there was another meeting here of an overlapping topic, but then I decided I should not miss this opportunity because this is a wonderful place and it is organized by one of the best bioinformatics departments in the country. It's actually interesting. Drug design and its translational aspects are practiced over a very long time, ever since chemistry and crystallography are being practiced, and there have been many successes based on this rational approach. But real breakthrough, it actually came in around year 2000 when the human genome in its entirety was sequenced, and people thought it is really a great opportunity. We can do anything and everything with regard to what works in humans as well as other organisms, and host pathogen interaction can be studied so well that you can actually discover many drugs automatically, whether it is for cancer or diabetes. But that proved to be a great deal, a sort of a dampener. Nevertheless, that wave in year 2000 actually turned a lot of people towards working in bioinformatics. And however, this department has sustained its interest for all through this and many other departments. So bioinformatics in India actually has a continuous steady interest. In fact, ahead of most of the researchers in India. In fact, it started with G.N. Ramachandran establishing molecular biophysics studies at Madras University and later in Indian Institute of Science, Bangalore. And that tradition continued, and we have had substantial leads to the extent that we have been a force to reckon with as a country. But real major discovery has actually happened, which can really fuel great deal of possibilities in drug discovery and its translation in terms of what we now have is almost automated structure determination of proteins. The first such program came in last year, what is called as AlphaFold, or there are many other versions of it, in terms of being able to computationally correctly predict three-dimensional structures of proteins. And there are algorithms that are evolving which are slowly able to discover which site in the macromolecules a drug can bind, and understanding whole of biology. Real breakthrough actually came almost 25 years after what genome sequencing worked out, and it seems to have a great deal of impact. And what fruits of it we are yet to see in totality, but this discovery was so big that within a year couple of people who were responsible for making this discovery were actually awarded Nobel Prize. So we hope that the drug discovery and its translational impact would be actually seen in years to come. And we have full strength in terms of what to do. And this discovery happened primarily for two reasons. One is while three-dimensional structures of proteins are being determined for almost 50 years or so, what is great thing about those of us who have been practicing structural biology is that we all have been collating and putting together all this data in one place in what is called as a protein data bank. There is a systematics of it, and everybody's data is uniformly put together. While we were ahead in terms of X-ray diffraction, but through NMR and cryo-electron microscopy, also many structures have been determined, and all of that has been collated. So most important input for being able to make predictions correct is the development of a database. Second important thing is actually both hardware and software in computation. And evolution of artificial intelligence has actually made this combination work in such a way that in last about 10 to 15 years, it actually became real that you understand all the rules of structure and you apply them to be able to develop new three-dimensional structures simply based on the sequence became a reality last year. So with this, bioinformatics is going through its second transformation, and drug discovery will get more focus, and hopefully this field will grow to greater heights. With this, let me close my inaugural address and thank you for your attention.
Thank you, sir, for your inspiring words. Now I request Professor Sanjeev Kumar Singh to address the audience.
Honorable Vice Chancellor of this esteemed university, Professor Dinakar Salunke, former Director ICGB, my colleagues on the dais, Dr. Dinakar Rao, Dr. Krishna, Dr. Tiwari, and Dr. Mohan, dignitaries of the dais, Professor Ramasubbu, Dr. Shekhar, my other scientific friends, Dr. Sudhakar, Dr. Hitesh Patel, all the BITS members, students, participants, very good morning to one and all. This is a great privilege and honor for me to stand here before to tell something about the Bioinformatics and Drug Discovery Society. The BITS serve as a dynamic platform for the scientific exchange connecting the field of bioinformatics, computational biology, and drug discovery with the broader landscape of life science. The society was established in the year 2017 with the vision to promote the advanced modern approaches to rational drug design through the transformative power of bioinformatics, a discipline that has today become the indispensable in biomedical and pharmaceutical research. It gives me immense pride to acknowledge the visionary leadership and the guidance of our founder, former president and mentor of the society, Professor T.P. Singh, currently INSA Senior Scientist at All India Institute of Medical Sciences, New Delhi. His foresight and commitment to advancing structural biology and bioinformatics led the foundation for what BITS stands for today. Currently, the society is led by our president, Professor D. Velumurugan, former professor and head department of biophysics at University of Madras, and currently serving as a member secretary of TANSI, Government of Tamil Nadu, whose outstanding contribution to the structural biology to continue to inspire the next generation of scientists. Under the guidance, BITS continues to grow as a national and international platform and that unites researchers, academicians, and students to share ideas and explore collaborations and drive innovation in computer-aided drug design. I fully agree with what Professor Salunke told. The contribution of bioinformatics and the contribution in the area of drug discovery is very high, and slowly it is moving leap and bound. So on this wonderful occasion, I would like to invite Professor Salunke to kindly confer the BITS Lifetime Achievement Award for the year 2025 to Professor Prakash Babu, Honorable Vice Chancellor of Pondicherry University, Pondicherry. So, I give you citation, please. And I request all the dignitaries to kindly join along with Professor Salunke.
Very good morning, all of you. Vanakkam. Namaskar. Dignitaries on the dais, Professor Dinakar Solanki, former Director of ICGEB, New Delhi, Professor Sanjeev Kumar Singh, Secretary General, Society of Bioinformatics, Professor Krishnan, Head of the Department of Bioinformatics, and he is also the instrumental in this organization, the conference, and Professor Dinakar Rao, convener of the conference, Dr. Mohan Kumar, and the Dean in charge, Professor Kumar, Basant Kumar, colleagues sitting of the dais, faculty members of the bioinformatics and other departments also I can see, librarian is here, and other colleagues are here. And the invited speakers, guests, and the conference delegates, students, research scholars, other invitees, and the media persons here. It's my great privilege to be here on this occasion. I am deeply honored by the Society, Bioinformatics and Drug Discovery Society, which they have considered my candidature for the Lifetime Achievement Award of this 2025. I thank all the Society office bearers and also the committee which has chosen me to this prestigious award. I am deeply honored. The conference, whenever I come to the bioinformatics or the biotechnology or the life sciences, always I feel that it's close to my heart because I also belongs to the same species, always. And Professor Dinakar Solanki rightly pointed out how the bioinformatics is being growing and still further it is growing. So during the early days of the research, during especially the drug discovery, when the drugs are synthesized and given it to the biology people, so we do in the in vitro studies, so then we go for the in vivo studies. Since I'm also associated with the pharma industries at Hyderabad, they say that 1 or 2 molecules from the 10,000 molecules they initiate will go to the clinical trials. There it may fail also. But the bioinformatics made their lives very much easy because before going to the wet lab, so earlier when we go for the any committees, when they wanted to propose for the animal studies, we used to ask them, have you done the in vitro studies? Because you cannot sacrifice the animals without proper purpose. So they have to do in vitro. Now we keep asking, have you done the bioinformatics studies? Unless you have the raw data that yes, this has the value in particular disease, you can go ahead, then you cannot go ahead with the biology studies because it is a costly affair. And also you cannot make a hit and trial with the molecules in the drug discovery. This bioinformatics actually changed or transformed the complete scientific research or the drug discovery or the health sciences completely. Now, the Professor Dinakar indicated that the second revolution is going to ahead. And even in the biology also, there is one more revolution is coming. That is the AI rats. Recently, that they have predicted that we don't have to do anything. Then, if you have the AI rat, you can predict that how the drug is going to affect the animal, or whether it is really going to work out or not. So, this is the tremendous change in the scientific field that it is growing now. So, when it comes to the bioinformatics, once you go for the molecule, and it's a lock and socket. So unless you know the molecule structure, size, whether it is fit into the receptor or not, so it will not work out. So if you leave it in the system, in the biological system, it's a ocean. If you do the in vivo studies, you are just keeping a molecule in the ocean. We do not know where it goes, how it goes, unless you have a pinpoint drug delivery where it goes and binds to a particular receptor. So that information is going to get from the bioinformatics studies. So these bioinformatics studies are very much inseparable and very much essential for the life sciences or the drug discovery. So I really congratulate the organizers, convener, and the organizing secretary, and the society especially for thinking of having this meeting in Pondicherry University. I understand that Professor Dinakar Solanki said that it's the first time that he has visited. I think many of you also must have visited first time. Please do visit the university campus. It's a beautiful campus. It's a really green campus that we have been maintaining it. And it's an untouched fauna and flora are there. And also Puducherry itself is a beautiful place. You can go around and see the important places in the Puducherry. I think organizers will take care of it. Once again, I thank the society, and also I wish the conference a great success for the next few days. And I wish you all best of luck and thank you.
Thank you, sir, for an enlightening speech, and thank you for supporting Department of Bioinformatics. And now I request Dr. S. Mohan Kumar, Organizing Secretary, for the vote of thanks.
Good morning, everyone. On behalf of Department of Bioinformatics and the international conference organizing team, I'm happy to propose vote of thanks. First, I would like to thank our Vice Chancellor, Professor Prakash Babu, for being a constant support whenever we interact with him, and when we went for when he comes to the department or when we go to him, he gives new ideas how to proceed research or the administrative part. Thank you very much, sir, and also readily accepting to come over here to give the presentation talk. Thank you very much, sir. When I contacted sir, he readily said, okay, Mohan, I will come for giving inaugural address. Thank you very much, sir, for coming over. Among your busy schedule, you have taken your time to come over here. Thank you very much, sir. And I would like to thank the BID Society, President Professor Velmurugan and Professor Sanjeev, to seed this idea, okay, to conduct this conference, drug discovery conference at the international level. Both has been instrumental and also guiding us to run the program, organize the people. Okay, thank you very much, both for Professor Velmurugan, Professor Sanjeev, and also the society members. Thank you very much, sir. And I would like to thank our directors, Studies and Cultural, Registrar, Finance Officer, and other administrative officials in our university who has been constantly supporting us to send up the proposals, getting the financial things done. Without their help and getting the things done, it's very difficult to organize such a big meeting. Thank you very much for them to get the things done. And I would also like to thank the international and national speakers. We have around 25 national speakers, and we have 4 international speakers. Professor Narayana Subbarao is here, and Dr. Shea is here. Thank you very much. And also we have several national speakers. A few of them will be joining online, but majority of them are here. Thank you very much for them. And also we have industrial scientists, particularly the software, we have from Advent, Zastra, Schrodinger, and Molsoft. They are here. Some of them are going to give a talk, and also there is a workshop that is scheduled. Thank you very much for them to be to come over here and train our students to use the software which is at the, typically used at the industry level. Okay, so it will be helpful for training our students, masters and PhD students, so that they can go to the industry directly from there. Thank you very much for them. And also I would like to thank DBT for funding. So without funding, such conference is not possible. So I would like to thank DBT for their funding. And I would like to thank EMRC and PRO section for providing the media coverage. Thank you very much. Also, I would like to thank the electrical wing and the computer section for providing all the computer connections, all those things. Thank you very much. And also, I would like to thank our department HOD, our faculties, PhD scholars, MSc and MTech students. So without their cooperation and help, arranging such conference is very tedious and difficult. So they have been working very hard, particularly the PhD scholars. They have been working very hard till yesterday night so that everything could be arranged. Okay. Thank you very much for them. And finally, I would like to thank the participants who had come from different parts of India. Thank you very much.
Yeah, very good morning, all of you. This is my first talk on a scientific subject in this university. So I am also happy that I got an opportunity at this platform of the Bioinformatics Conference. I thank the department and also the society for giving me this opportunity. When the Professor Dinakar and all his colleague Krishna and everybody came, they said, "Sir, we wanted to honor you with this award." Then they have given me an option that you may deliver a talk, or that's if you don't want also, or if you are busy also, there is no problem at all. They have given me that option, but I don't want to take that option because this award should be for scientists, not an administrator. So I wanted to take that opportunity and to deliver a talk and show that something what is going on in the lab. Still, my lab is active lab. Still, I have the 8 students are there in the University of Hyderabad. They are actively working on the neuroscience area. So I would like to present some of the research data that we have generated earlier and also the currently. So as I said that my lab is working on the neurodegenerative diseases. And so I'm happy to be in the part of this international conference come workshop on the Global Summit on the Innovative Drug Design, Discovery and Translational Research. So we are using some drugs actually. So the University of Hyderabad Chemistry Department is kind enough to give us some of the drugs. We are screening against the different models. And also we are also using the repurposing of the drugs. Drugs already in the market, so they have the different potential, that also we are using it. So little part I am going to present. So today's my topic is the blood-brain barrier, guardian of the brain in health and diseases. So then I will go to the next slide. So these are the research areas that my lab has been working. This is the ischemic stroke. We use the animal models. We create the animal model for the stroke. It's called as the MCA occlusion. We occlude the middle cerebral artery in the rat, and we go for the studies. And also we also get the human samples from the NIMHANS, Bangalore, of the postmortem samples for the stroke patients, and we study with that. And there is epilepsy, we get the temporal lobe epilepsy, where actually surgically only, surgery is the only option for the epileptic patients. They remove some part of the epileptic seizures part, and that we are using for these studies. So the other one is the sciatic nerve injury. So this project has been evolved actually by the LAPRA India, when they wanted to have to study the leprosy patients, where their nerves toes nerves it starts from the degenerating. So therefore they lose the sensitivity, and that's how they lose the digits, fingers and also the digits of the legs. So we have got a project from the ICMR and we started working. So we wanted to study for whether they can be regenerated or not. So for that we need an animal model, and we have to see how that can be mimicked or replicated. So that's how we developed this animal model, where the sciatic nerve can be ligated partially or completely. So that initiates the degeneration of the myelin sheath. Once the myelin sheath is degenerated, so the nerve will lose its actual potential, and so that it cannot transmit the signals. So that's how we developed it, and we have been working it, and we published some papers also. The other one is the glioma. Glioma is the deadliest form of the brain tumors, as everybody knows. it will not give the, once the patient is diagnosed with the glioma, he will not survive more than 11 months to 14 months. That is the maximum period. So this is what I keep telling to people, especially the young students and the research scholars. If anybody can identify or discover a drug which can treat the glioma patients, definitely they will get the Nobel Prize. So now that is such a kind of research is required from all of you. So this is another one. So other one is the cerebral malaria. You say cerebral malaria is, the malaria is a poor man's disease, nobody works on that. So therefore we have taken up that one, and that is another model that we have. So we have I was trained in Japan in a hospital, stroke hospital that is, for the stroke research. After coming back India, I have started this one as well as cerebral malaria also. So because both are actually more or less the same thing. In the case of the stroke, cerebral ischemia, it's a kind of occlusion that goes to the clots that is formed in the heart that we transported to the brain, and where it blocks the particular artery, especially the middle cerebral artery, where it goes and blocks that particular region, then the person will get paralyzed on the other side. Left hand and right leg, left leg will be paralyzed if it is occlusion on the right side. If it is occlusion on the left side, it will paralyze on the right side. If somebody got paralysis on the right side, right hand and right leg is paralyzed, he will also lose the speech. He will also lose this because left side is the speech center is located in the brain, therefore he loses the speech also. So that's all. And in the case of the cerebral malaria, so that is the focal ischemia, that is one particular part is degenerated. And also there is one more ischemic condition called as the transient ischemic attacks. That is sometimes it comes the ischemic attacks and there won't be any clear infarct, but some neurodegeneration will be there. So therefore that's called as the transient ischemic attacks. That mimics the cerebral malaria. Cerebral malaria mimics the transient ischemic attacks in the human beings. So that's how we have developed. And this is also more or less similar to that of the Japanese encephalitis. So that's how we are. So now I'm going to present some part of this work which we have been doing from my lab. And before going that one, let me talk about something blood-brain barrier. So students may not be aware of it, what exactly the blood-brain barrier. So there is no physical barrier, but it's a blood vessels. So there are two organs in the human body, blood vessels are completely different. One is the brain, the other one is the kidneys, where the filtration is there. But the blood vessels are completely different, both are in opposite direction in the kidney as well as in the brain. In the kidney, it requires the filtration where the blood vessels or endothelial cells are actually loosely packed so that the urine can be filtered. But whereas in the case of the brain, they are completely packed. It will not allow anything to come out or go inside. So that is called as the blood-brain barrier. So this blood-brain barrier actually protects the brain from several things. So this is the blood vessel, this is the endothelial cell. And these are the brain cells. So this is the lumen of the blood vessel. You can see these are all called as the astrocytes. For the sake of the students, the brain will have the two types of the cells, neurons and non-neuronal cells. Non-neuronal cells are called astrocytes and the neurons. Astrocytes are called as the soldiers and neurons are called as the kings. So king is always protected by the soldiers. Soldiers will die, soldiers will sacrifice their life, but not the king. So these astrocytes are actually like a soldiers. These soldiers actually, they keep their end feet towards the blood vessel. So any blood that should go to the neuron should go via the astrocyte. So if there is any toxicants, every day we are all eating a lot of toxicants, especially the students when they go out of the shop or pizza market or drinks, whatever it is colored, it has the toxicants. You don't believe that one, they have the toxicants, but these toxicants, they don't enter into the brain because of the blood-brain barrier. Any colored substance that you are eating, it will not go to the brain. That's how we are all saved, because of the blood-brain barrier. So this is the duty of the astrocytes. Now the blood-brain barrier has several important functions, like it protects the brain from the foreign substances in the blood that may injure the brain. It protects the brain from the hormones, neurotransmitters in the rest of the body, and it also maintains the constant environment for the brain. So this is how effective security system is an effective security system. This is the blood vessel, these are the astrocytes, these are the neurons, and this is how the cerebrospinal fluid and other areas will be there. So that's how it protects the one. It's a specialized endothelial cells are there in the blood-brain barrier, and it's a intraluminal portion of the brain capillaries. It's a semi-permeable capillary, it will not allow everything. And blood-brain barrier separates the brain and cerebrospinal fluid from the blood. Presence of tight junctions is like a zonula oculodense. This is one of the important proteins that protects the blood-brain barrier. So that also I will talk about this one. Between the continuous cells and the absence of the endothelial pores, there won't be any pores in the brain. Now, if you see this one, this is the blood vessel. So these are the astrocytes, and this is the neuron. So neuron will go and attaches to the astrocyte, not directly to the blood vessel. So if you take this one as the picture, you can see there are several astrocytes are there, microglial cells are there, pericytes are there. So many are actually attached to that. So blood vessel is not directly attached to the neuron. Neuron will not directly contact with the blood vessel. And these are the small proteins that they are actually plays an important role in the stabilizing the blood-brain barrier. So at the endothelial cells, these are the proteins, some of the important proteins that play an important role. Now, if you take this is the healthy area of the blood-brain barrier, and this is the diseased condition, you can see that healthy area, it will not allow anything. It will not allow anything, including the immune cells. As you know, the brain is immunologically silent. Brain is immunologically, no immune cell will enter into the brain unless blood-brain barrier is damaged. So immune cells are like though they are protective in nature, but if it goes into the brain, this also indiscriminately kills the cells which are infected and the non-infected. So that's a different work that I will present that one in different place. Then this is the diseased condition where the meta-rheumatoid proteins are actually damaged, and also ICAMs are elevated, and this is the place where the immune cells are actually entered and reactive oxygen species are actually generated. So these are all the diseased condition. This how makes the blood-brain barrier permeable. Now, what are the diseases associated with? If you know the multiple sclerosis, where the blood-brain barrier is damaged, and also in the case of the stroke, in the case of the Alzheimer's disease, epilepsy, traumatic brain injury. These are all the places where the blood-brain barrier is compromised. Once the blood-brain barrier is compromised, brain is actually open for all the toxins. That's how the damage takes place. Now, this is the one actually I would like to touch upon now. This is the area. This malaria is the rampant, especially in African countries. In India, it is actually now located only in some pockets. It's in the interior places of the forest areas, like in the Orissa, some part of Telangana, Chhattisgarh and all. So some of this malaria, when it goes to the different level, that's called as the cerebral malaria. These cerebral malaria cases, it will not come to the light actually, because there are all people in the hamlets or forest area. So normally they silently suffer. Only primary health center, they will treat only for the malaria but not for the cerebral malaria, because they do not know the implications of the pathology of it. Now, this is actually the Plasmodium falciparum is the one which is actually culprit for the cerebral malaria, and also nowadays the Plasmodium vivax is also part of this disease. Now, this is I am lucky to see the building of the Ronald Ross in Hyderabad, Secunderabad. He is the one, he discovered the Plasmodium, the malaria parasite associated with the mosquito. So, opposite to this building there is one tank called Tank Bund. He used to go to that place for a walk, and he used to catch some of the mosquitoes every day, and he used to dissect, and he has a small microscope, small compound microscope, still it is there, kept there in the building, and he used to dissect the gut of the mosquito. Then we saw some of the nodules. So that's how the great discovery led to the Nobel Prize. And these are his own writing paper, where he pointed out that these are the nodules. He has seen some of the nodules in the gut region of the mosquito. So this is how the malaria research has been initiated. Now, simply I would like to talk about the cycle. I will not go complete cycle because people must be aware of it. Now, my interest is actually, once the infection is mosquito bites the human being, the sporozoites goes into the circulation. It goes to the liver stage, then merozoites, then it goes and enters into the RBC. Here actually my pathology or my study starts from the how the blood cells are actually infected and what has happened to that one. So once this is actually enters into the RBCs, they eat away complete RBCs, they burst open and go and invade the other RBCs. So it indiscriminately kills all RBCs. Now, so for the sake of the students, this is the RBC. Parasite enter into the RBC, then it goes and eats the protein part of the hemozoin. then it multiplies and it bursts open and it goes and infects the other places. So see the beautiful architecture of the RBCs. It's like a coins. Now, when the parasite is entering, as you all bioinformatics people, it cannot enter straight away. It will not. So it has to make some contact. It has to make some contact. It's kind of it has to attach us to the receptor where it can bind and where it can be allowed into the RBC. So therefore, it knocks around the RBC. It knocks around the RBC. Parasite knocks around the RBC. Then it finds the receptor where it can be received. Then it enters into the RBC. So during this process, it damages the surface of the RBC. This is how the pathology of the cerebral malaria initiates. Once it damages the RBC, beautiful architecture of the RBC is damaged now. Now it has become like a sticky. It has become like a sticky. Now apart from that, parasite also, RBC doesn't have the nucleus, but the parasite synthesizes some of the proteins. These proteins are transported onto the membrane of the RBC during pathology. These proteins on the membranes are given or made arrangement, some kind of arrangement is called as the host pathogen reaction. These proteins makes the parasite to evade from the immune system. This kind of arrangement is being done by the parasite. So parasite is actually very intelligent than us. So whatever the drugs that you make it for a particular antigen, it changes its structure. That's all. So that's how the malaria has become. Still, it is not eradicated. Actually, Indian government, they started the malaria eradication program in 1967. But later, they changed malaria eradication program in 1980s as the malaria control program because they realized that it cannot be eradicated. So even now, it is going on. So that's how the parasite has become resistant to the existing drugs. Now, once the surface is damaged, it becomes sticky. Once it is sticky, the infected parasite, infected RBC, is stick to the non-infected RBCs. That's how it forms the clots. These are small clots are formed. Once these clots are transported to the brain, that's how the cerebral malaria starts. So this is one condition. Now this is the newspaper from the Andhra Pradesh. I could get it. This boy actually he got severe malaria. So then malaria has been treated, but that boy stopped talking. He lost his speech. He also lost his walking. So this is actually precisely the cerebral malaria condition. This is. So how the doctors or the primary health center people at the rural areas, they treat the malaria, that's all. But it has the beyond their control. And this is the actual pathology. So this goes and binds to the endothelium, it binds to the clots, and also platelet derived particles are released from the parasite, and also blood-brain barrier is damaged, and release of the pro-inflammatory cytokines is another. This is a cytokine theory, that's another one. And also endothelial activation is also releases here, that also damages the blood-brain. So these are all the changes that occurs in the blood-brain barrier by the parasitized RBCs when they attaches to the endothelium of the microvenules of the brain. The whole pathology starts, the infected RBCs are transported to the brain, where they attaches to the endothelium of the brain, where it interacts with the blood-brain barrier and it neutralizes or damages. So I got one paper, interesting paper in 2001. This is how we started cerebral malaria research in my lab. 2001, you can imagine. Okay, so almost 25 years back. So this paper said that is the ischemia involved in the pathogenesis of cerebral malaria. So people say that cerebral malaria, people die of the because of the hemoglobin is reduced because of the RBCs are being damaged. But beyond that one, so this paper says that there is a ischemic conditions also prevailing in the brain. So there is a ischemic conditions are prevailing like a stroke. So what is happening there and all? So that's how the research has been we started in our lab. So this is the MRI of the brains of the people. This is the normal brain. So this is the cerebral malaria infected brain, where you can see the edema. Edema is the first one. Inflammation and edema is the first one to kill the patients. Now the research here. So we inject the parasites into the rat mice model into the tail vein. So then it develops the malaria. From 5 to 7 days or 5 to 9 days, it causes the cerebral malaria. By 11th day, it will die. So this is the actually scenario, and there we study at different stages. So we see the sequestration, IRBCs, leukocyte infiltration, and also hemorrhage of the brain, and also blood-brain barrier dysfunctions. These are all the parameters that we look into that one. So these are the mice. So this is infected with so this is control mice. This is the cerebral malaria-induced mice, and this is the malaria. So once we have to say that this is cerebral malaria, we have to minus the malaria. Unless you minus the malaria, it cannot be cerebral malaria, right? So the Plasmodium falciparum, so this is the Plasmodium berghei anca, which is similar to that of the Plasmodium falciparum, and this is the Plasmodium yoli. This causes only malaria. It will not cause the cerebral malaria. So that is our positive control. So now we have the control and also positive control, and this is the cerebral malaria, where normally the cerebral malaria-induced mice are being studied for their postural stability and symptoms of the neurological symptoms by using one scale. And now we have to show first the blood-brain barrier, whether it is damaged or not. That's because our story starts from there. So we have to show that the blood-brain barrier is damaged. So all the students, as I said earlier, the colored sweets that you are eating, that color will not enter into the brain because your brain is intact with the blood-brain barrier. Same thing happens in the case of the mice. You just inject some kind of blue color or blue dye, it will not enter into the brain. It goes into every organ. It goes into every organ, but it will not enter. See, this is the mice. Once it is injected the Evans blue, complete mice become blue in color. But once you open the brain is pinkish in color. It did not enter into the brain. So this indicates that the blood-brain barrier is intact. So it did not enter into the brain. So now if you see the cerebral malaria cases, this is the malaria. So once there is a malaria is induced, fur is raising. That's called, it's a kind of a symptom for the mice or animals that it's not healthy. It has some disease, especially the fever that indicates the raising of the fur. Now you can see the brain is blue color. So this clearly indicates that the blood-brain barrier is damaged in the case of these animals. Now our story started from here. Now, so we have to go for this is called RMCBS. It's a rapid murine coma and behavior scale. So this is similar to that of the stroke patients also. We see the sum of the parameters and we estimate that one, whether patient, what is the length of the, what is the depth of the neurological symptoms that he has, that one. So this is the RMCBS is the one which we measure before taking the animals. And this is the scale, actually it's a 20-point scale, 10 parameters are being given. Now we also study the cognitive abilities of the animals. So cognitive abilities we compare with the control, and also we study for the cerebral malaria mice. So we study the various parameters. It's a spatial learning, and for the memory we study this T-shaped maze where we keep the food at one place and we train them, and the normal mice, wherever there is a food is there, straight away it goes there. But if it is mice with the cerebral malaria and it's the neurological symptoms they initiated, it cannot go and identify that one. And other one is this is another is for the novel object recognition. So we keep at different chambers are there. It has to go and explore the place and all. So it will be trained for some time. So if it is trained normal mice, it is straight away it goes to that place. But cerebral malaria one, so that causes the retrograde amnesia by using this test. And also the other one is the bout type maze test. This also indicates the anterograde amnesia. Now, so these are all so we are proposing finally some drug, that is anti-malarials are being given to the people, those who are having. And along with the anti-malarials, you should give some drug which can protect the brain. So if both are given, so some of the children, those who suffered in the early stages of their childhood, they suffer the cognitive impairment at the later stage. So that indicates that during the malaria, he must have got the cerebral malaria, and that must have been unnoticed. Therefore, there is a cognitive impairment that continues for the entire life period. So therefore, the patients or the children especially should be treated for the malaria as well as the cerebral malaria. If you give a drug for the cerebral malaria also, its lifelong protection will be there. It will not affect the brain. So anything affects the brain, it's a long term. It's neurons, once they die forever. It will not go further. So it will not be regenerated. Therefore, we are proposing that there is a minocycline. Once we used minocycline, it protected the blood-brain barrier. Minocycline up to some extent. So we have to use it's called as the combination therapy. combination therapy with the antimalarials as well as the drug that can be used for the protecting the blood-brain barrier. Now, with this, I will present the small part that we have done. So, once we know that these animals are having the cognitive impairment, so the memory loss is there in the case of these animals. So, what is happening actually? What exactly is happening to these animals for the memory loss? As you know, the synapses is the one which is the synaptic density. Synaptic density is the one which determines the memory or cognition. So, once the synaptic density is lost, that means synapses are lost, and also synaptic cleft is lost. So, once these are actually, number of synapses are directly proportional to the intelligence or whatever it is, memory or the cognitive abilities. Once this density is actually reduced, then it will have the impairment. So, now we have studied this, these are the neurotrophic factors. Neuronal arborization. So, neurons actually, once there is a cell body and it has a lot of dendrite and dendritic spines. Each spine actually, so Shastri, how are you? So, each spine actually it's a one synapse. So many synapses are there in the case of the neuron. So, once those synapses are actually reduced, that directly affect the ability of these animals. So, now we have studied how the synaptic transmission and the long-term potentiation, and finally it went into. So, this is actually this is the neuron. So, this is the axon, and these are the dendritic spines. Each dendritic spine is this one. Each dendritic spine is nothing but one synapse. So, these synapses actually being controlled by the lot of proteins like cofilin, and also some of them we have studied. So, these are the MAP proteins. So, these are all. So, to check that one, what exactly happening to these animals, what is happening here. So, if you say this in the case of the dementia patients, you can see that the arborization is not complete. There are some vacuolation are there, not completely formed, this one. And also these are all in the stroke patients, you can see such a certain of these neurons, not continuous. So, these are all the actually effects of the neuron where it determines the cognitive abilities of that particular person. So, when we study in these animals also, we found the similar condition here. You can see these are all the vacuolation where actually the synaptic connections are lost. So, these are all the completely, so this is the neuron where completely is lost. And so, these are all the, once in a different animal, these are all the actual controls. You can see the control, you have the complete density of this one, but whereas in the case of the cerebral malaria, you can see they're all actually thinly scattered and it has actually damaged the arborization and all. Now, so using the shoal analysis is the bioinformatics tool where you can analyze how this arborization is affected in a different circles. So, different places where from the initial stage of the dendrite or the synapse or later stage of the post and pre-synapse also you can study using the shoal analysis. Now, these dendritic varicosities were observed in the neurons of the cerebral malaria brain through the Golgi complex staining. Spiny density neurons were significantly reduced in the case of the cerebral malaria, and also dendritic arborization pattern has been analyzed by shoal analysis with a less in the case of the cerebral malaria in groups rather than the control ones. So, this is our finding. So, we have published this one in the Annals of Neurology, which is a medical journal. They publish only one paper per journal, basics research, otherwise they are all. So, we could publish that one. So, I only truncated actually a lot of data is it, I truncated this one. So, other few parts are there. So, when we come to the treatment part, what can be done with this? So, we identified that this is epocyanin. Epocyanin is the natural organic compound has been isolated from variety of the plant sources. So, we have used this epocyanin as the adjunct therapy along with the antimalarial so that this can help the patient. So, you can see this one, once we have used the adjunct therapy, so they are all actually, some extent it is restored. So, therefore, we propose that the malaria, some children especially admitted in the hospital should be treated with the antimalarials as well as the adjunct therapy which can protect the brain. And also minocycline also being used. This minocycline also reduces the hemorrhages and also the neurodegenerative places where it is having some parts of the neurons are dying. So, minocycline also can be used as the adjunct therapy. And so this is the fluorescent stain. This determines the number of neurons are actually they are dying. So upon the treatment with the minocycline, number is reduced. Now the conclusion that the dendritic varicosity has been reversible through the treatment of novel nootropic drugs as adjunct treatment, and apocynin effectively worked in the recurring the mice from the CM, cerebral malaria, prevented formation of the dendritic varicosities. So now we have the this is a polycyclotol derivatives also we were given by Professor Govardhan Mehta's lab at the University of Hyderabad. So they have been working with these different derivatives and all. We are fortunate to have that one. So we wanted to use along with the artemether. Artemether is the anti-malarial. We wanted to use whether this combination can also really helpful. So we have studied the as cognitive tests, and we showed that the cognitive effect is there. And when we used that one, it's ratios. See. So this is the cerebral malaria, and this is the ARM, the anti-malarial, and these are all the drugs. So once these drugs are actually given, so there are actually 2 and 4. Actually, they are really promising. They are really promising. So this is actually restoring the cognitive effects of this one by using the combination therapy of this particular drug. And we have even studied the phosphorylation. So this drug is, so once we published in the Annals of Neurology, they also raised an issue that whether this has any relation with some of the proteins associated with the Alzheimer's disease. Then we wanted to check with this one. So when we checked the tau proteins, so tau one is phosphorylation and dephosphorylation of this tau. So that also affected actually, and this drug is actually restoring that one. So we are proposing that. So this has some of the cognitive dysfunctions, even on par with that of the Alzheimer's patient is some part, little part, but they can be restored by using the drugs that we have used. So this drug is actually, we have published this paper recently, very recently, with Pondicherry University as my affiliating place. So, yeah, other small part is there in another 5-minute cycle work. So the beta-dystroglycan is the component that dystrophin glycoprotein complex. This alters the astrocytes. So this is the actual, once the astrocytes are altered, automatically the blood-brain barrier is damaged. So this disruption causing the edema in the case of the cerebral malaria animals. And so this is the dystrophin. Actually, this is the one actually beta-dystrophin glycan. Actually, this actually regulates the complete proteins of which are associated with the integrity of the blood-brain barrier. So this is present in the astrocytes and feet, regulating the blood-brain barrier integrity. It's a part of the dystrophin glycoprotein and forms a structural linkage between the extracellular matrix ECM and also the alpha subunits that the skeleton through the transmembrane protein. And also beta-dystroglycan cleaved by the metallomatrix protein 9. So that actually culminating into the vasculature damage leading to the edema. Now, once we have studied that one, it's shown that it has been damaged, so by the RMCBS one. And these are all the controls we have used. This is the day 5 of the cerebral malaria animal. It's normal only, but day 7 it is infecting the cerebral malaria. So then when we used some of this is the non-cerebral malaria, when we used the artemisinin, so it is reduced it. And edema is also reduced here by using the artemisinin. And also some of the proteins that they are, so JEDVA1 is the one which is actually plays an important role in keeping the integrity of the blood-brain barrier, and also the alpha-synuclein and laminin. These are all shown to be, in the case of the day 7, they are all altered, but whereas when we use the artemisinin, so they are all being reduced partially, if not completely. So therefore, not only the anti-malarials, we should also use the adjunctive therapy. Now, these are all the immunohistochemistry of the ones. Day 5 and day 7 is the actually highest one, but whereas the use of ARM, artemisinin. So, we always use the hippocampus and the cortex. Hippocampus is the one which actually it affects the memory. Memory of the people will always be impacted by the hippocampal region. And this GFAP actually, this gives the astrocyte and feet. So these are all the astrocytes at different. So day 5 and day 7. So this is the control one. You can see the arborization and all. Whereas after giving the ARM, so this is restoring to the normalcy. And so this is the GFAP histochemistry. You can see that one in the Burgess patient, that where how it is getting affected and also how it is going to restore. And also this one with the beta-dystroglycan also we have shown. And also here also the ZWO1, how it is getting affected here in the laminates and all. So that is also shown that the day 5 and day 7 is the one which is highly affected. But whereas upon the ARM treatment, so that has come to the near to the normalcy. And the CD31, which is the marker for the microglia or the reactive microglia also being elevated in this, but upon the treatment that has come down. And also the IBA1, so which is actually this is the microglia, reactive microglia actually releases some of the proteins that they are actually responsible for the inflammation in the brain. And again a GFAP. So this is the aquaporin. Aquaporin is the one actually this causes the normal condition. Once the disease occurs, these are all actually destabilizing the neurons, astrocyte end feet, and also the blood vessel, where it causes the release of the water and it causes the edema in the brain. So this is the one actually playing an important role here. And that is also being restored by using the ARM. And so we studied the VEGF, vascular endothelial growth factors. That's also we have studied, and in the hippocampus and the cortex, where it showed that one. So this is the day 5 and day 7. Day 7 is the maximum effect is there, whereas upon using of the ARM, it has come to the normalcy on par with that of the control one. And so this is the basement membrane degradation. Basement membrane degradation is seen in the case of the highest in the day 7, but whereas it is reduced or restored upon the ARM treatment. And this is CD31 staining intensity indicate the endothelial cell interaction with the extracellular matrix, where the extracellular matrix plays an important role in the endothelium for the damage as an indicator for the damage of the blood vessel. So the conclusion, so these are all the ones that even before the onset of the disease symptoms, changes at the molecular interaction at the cliovascular level interface is the one to be addressed. And also beta-dystroglycan cleaves by the MMP9 led to the disruption of the dystroglycan protein. And also astrocyte alterations are the one which actually responsible for the loss of the integrity of the blood-brain barrier. So this paper is recently published, it is accepted now. So again with the Pondicherry University as a co-author. So thank you. So these are all my research group, and the data has been presented by Shailaja and also the Praveen Kumar. These two people contributed for this paper, and here my wife is also here. She always supplies the drugs from the chemistry, and where we screen several drugs in the. So thank you very much, and I thank the funding agencies, DST, DBT, and ICMR. Still I have the project, ongoing projects are there. I have to handle that. And I profusely thank the Society for the Bioinformatics and the Drug Discovery Society, and also the Bioinformatics Department and the organizers. Thank you very much.